This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 705, recorded on January 8th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody, and hello, Vincent. And, it's cloudy outside. There's a little bit of a wind. There was a lot of wind yesterday, and the temperature is unseasonably warm. It's in the high 40s, I think. It feels like it's going to start springing into weather that we're not used to at this time of the year.
Dixon, I can see you across the Hudson. Can you wave to me? Hi, Dixon.
Wait, I'll give you my Queen wave. We've been watching The Crown on Netflix.
From Austin, Texas, Rich Condit.
Hi, everybody. Let's see here. 51 degrees, headed for 55, partly sunny. All good.
From Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. I want to, go to Dixon's because it's only 39 here, although it is a little bit more sunny.
We can get a heat bubble from New York.
We can wait if you want to drive up and join him.
Sounds good. Perfect.
And from Western Massachusetts, Alan Dove.
Good to be here. It's clear blue skies, temperatures 4 Celsius, which is about 35 or so Fahrenheit, and it's been the same weather all week long, just clear skies, low to mid-30s.
Our guest today is from a little bit west of Rich Condit. A little bit. That's my northeastern view of the U.S.
She's even west of Western Massachusetts.
She's the scientific director in the Colorado Department of Public Health and Environment. Emily Travanty, welcome to TWiV.
Thank you. It's a pleasure to be here.
And are you
Emily, is that in Denver?
Denver. Yeah.
Yes, I'm here in Denver, Colorado, at the Colorado Department of Public Health and Environment's State Public Health Laboratory.
Okay.
And
so you're at an altitude of about 5,000 feet?
About 5,000 feet, and I'll report that it is 43 and sunny here today in Denver.
Nice.
Very good. And I guess you're at work because you have some official-looking documents on the wall, right?
Yes, I am here in the lab today at work.
Very good. We have Emily on to chat about their detection of VOC 2020112/01, which I've been told not to call by its geographic site of origin. I was yelled at the other day. So, VOC.
Wait, so is this the same as B117?
Well, yeah, it's the same thing. B1117 is actually the lineage, right?
Right, okay.
So you could use that too. That'd be fine.
Because VOC could be volatile organic compounds. That might get a little confusing.
Variant of concern.
Emily, what do you call it?
We actually have been using the B117 because it's a little bit shorter and making it a little bit less of a mouthful to talk about.
Okay. B117, just 2 ones? Mm-hmm. Okay.
2 ones.
That works for us, right?
Yes. Yes.
Good job. Mm-hmm. All right. Before we talk about science, Emily, tell us a little bit about your background. Are you from Colorado originally?
I'm actually from Wisconsin originally.
Mm-hmm.
I grew up in Wisconsin and did my undergraduate at the University of Wisconsin in Madison, and then came to Colorado for graduate school and have not left since.
So did you interact with any of the virologists in Wisconsin?
Not really. I didn't really get into virology until graduate school up at Colorado State University is where I moved from. My undergraduate work was in microbiology and genetics.
So I did a Zoom a couple of weeks ago with the University of Wisconsin Microbiology Club. Mm-hmm. And they just sent me a Wisconsin Microbiology t-shirt, which I didn't have. It's great. And they sent me
they're going to badger you to wear it, right?
Right.
Hey, you bet. They sent me a milk, what they call a milk glass, which is a beer mug, right?
In Wisconsin.
Joking about it. So at Colorado, what did you work on?
So in Colorado, I was working at the with the arthropod-borne infectious disease group, and I did my graduate work on dengue virus with Carol Blair and Barry Beatty and Ken Olson up at CSU.
We just had Carol on TWiV a few weeks ago.
Saw that one.
It's a ASTMH episode.
Mm-hmm.
And we had, tell me the name, I'm forgetting, also from Fort Collins. He does the deer mouse SARS-CoV-2 guys.
Tony
Shounts.
Shounts.
Tony Shounts. Sorry, Tony. On as well. Yeah. A lot of Colorado virology. It's great. So you did a PhD in Denver or Anschutz, actually, right?
It was actually in Fort Collins.
Fort Collins, right. That's where
CSU.
Clara.
Mm-hmm.
And did you do a postdoc as well?
I did. I did postdoc in Denver at Anschutz. That was actually on coronavirus with Catherine Holmes. And that was right around the time that SARS had happened, was when I finished my graduate work. And I moved into respiratory viruses at that point and was working in the Holmes lab on looking at host interactions with receptors for SARS. Mm-hmm.
I visited Anschutz a couple of years ago, maybe in 2018.
Okay.
And
that was after I had been there. I was actually here at the state at that point.
So after your postdoc, you went to the state directly?
No, first I was at National Jewish for a little while working with Robert Mason on innate immune response to influenza in primary human lung cells for a while. And then from there, I transitioned to public health from the research world and came to the state. Been with the state for about 8 years now.
So that transition to public health, how did that happen? And does it involve additional training?
It did involve some training that I was not formalized training, but a lot of hands-on training and working with training that I could get through the Association of Public Health Laboratories. They have trainings available. And it was a big learning curve to come from the research world into public health, which is not at all a clinical lab and not at all a research lab. It's sort of a lab unto its own, in sort of draws a little bit from all of those worlds. And moving from focusing on one specific interaction of one virus and one host, and then coming into the role that I started with in the public health laboratory. I came in as a supervisor over clinical microbiology, serology testing, and having to switch to not focusing all day, every day on just coronavirus or just influenza virus, but to be asked questions about rabies and then syphilis and then enteric bacteria, and drawing from all of the various things that I've done in my career up to the point that I had gotten there was helpful. But it was a different mindset for sure to move to that.
So I'm curious how this happened. So did you think I want to be in public health and go looking for a job, or did the job come up and you said that looks interesting?
Well, I think it's a little of both. I knew I sort of wanted to do something that took my research background and moved towards something a little bit more forward-facing in interacting with the public and doing things that impacted people's lives daily. I thought about moving towards public health right after my graduate work. Interestingly, while I was at CSU is when West Nile was moving across the country, and so we had a lot of interaction in the arthropod labs that I was in with the CSU folks interacting with the CDC that was across the street from us at that time. And so I got a taste for sort of the idea of what happens on the national level as a new pathogen emerges in the country. And it was very interesting to me at that time. But I think my heart was still in the research world. So I took, postdocs that were more traditional research-based postdocs, and then the position opened up at the State Public Health Laboratory. And I saw that as an opportunity to draw on my background in virology and in, some of my undergraduate work as well into a new position here. And now in this role that I'm here, I get to draw on all of those pieces because, obviously I need the infectious disease background for the current topic of the day and the pandemic that's going on. But here at this public health laboratory, we also do enteric bacteria surveillance as well for foodborne outbreaks, as well as we oversee newborn screening here. So there's a little bit of genetics. So I'm kind of tying together all of my background pieces into the current role that I have in public health.
So, Emily, when you were involved in the West Nile outbreaks, we obviously were at the epicenter over here near the Bronx Zoo. And Tracy McNamara was a vet. And she tried to get that data actually looked at by people that had connections with not just animal-borne infections, but also human. So zoonoses versus human. How closely associated is the Colorado Department of Health, where you work, with the veterinary societies of, let's say, Colorado and other parts in the West?
So we do have connections with our veterinary colleagues. We actually have a state vet that's embedded in the Colorado Department of Public Health.
That's important.
Yep, and we do rabies testing here for animals across the state. Working also with our partners at CSU and their diagnostic lab as well. And there's hantavirus surveillance that still happens in Colorado.
Sure.
Yeah, so it's definitely an important piece of public health, is that sort of interaction between humans and the environment, and animals and the environment, and how they all overlap.
Right. Exactly.
So let me get the administrative structure. The Colorado Department of Public Health and Environment is the overarching association. Then you are head of a lab within it, right? Yes,
that's correct.
And the name of the lab again is
the Colorado State Public Health Laboratory.
Okay. So what does that laboratory do? You're the director of it, right? What are the things it does?
Right. So we have 4 major scientific programs within the state laboratory. The most forefront program right now, because of the pandemic, is my microbiology program, which is responsible for testing for foodborne illness, as well as infectious disease, sexually transmitted diseases, and tuberculosis surveillance. We do a lot of surveillance-based work. Most of the work we do is not primary diagnostics. Most of the samples that come in, we either know that they have salmonella, and we're trying to figure out if the salmonella in this patient sample matches the salmonella that was pulled out of chicken in my environmental microbiology lab, and then do molecular fingerprinting to see if they match. So we do a lot of outbreak investigations at the state level. I also have a chemistry program, which is primarily tasked with EPA-related regulations around drinking water and safe water, and also does foodborne chemical contamination in food. We have the newborn screening program, which screens all newborns in the state of Colorado and Wyoming for over 35 molecular or metabolic and genetic disorders in newborns. And then the 4th program that we have is a little bit unique to Colorado. We've stood up a marijuana or cannabis reference program to look at establishing some gold standard methods for how to test cannabis and hemp-related products for consumers. And we don't test all of them in the state. We're pretty much a reference laboratory for that. And we also have a large arm that does certification of other labs within the state. So we certify hemp testing labs and food and milk and water testing labs across the state.
And is that called the Rocky Mountain High program?
I think we should get that put on the shirts.
So when in 2020 did you start doing COVID-related things, and what were they?
Yeah, so it really started actually at the end of January of 2020, when the CDC was pushing out their first assay for COVID-19. I distinctly remember, though, seeing the emails come across December 31st of 2019 of a pneumonia of unknown origin. I remember seeing that come across and thinking, is this going to be something? Who knew we would be here today? And that first test was sent out early to state laboratories. Then we initially started working on trying to get that thing validated immediately. We wanted to be ready so that we could test anything within Colorado. CDC is a fantastic resource to send things to, but Colorado is pretty far from CDC. So whenever there's the opportunity to bring a new test on that we can do here in-house without having to have a delay for transport, we look rapidly to see if it's something we can bring on. Unfortunately, it ran on the same platforms we were using for our flu surveillance. So it was a pretty easy transition to try to bring that test on. And once we cleared all those validation hurdles, we thought we were ready and set to go with a capacity of about 160 tests a day. We thought we're ready. We were not. We rapidly had to increase from that point.
You have to add a few zeros to that 160.
We did.
What month did it start to increase? Right in February?
So it did. It started increasing in February. And I can pull up my numbers here if I could get my Tableau Viz to load real quick for me. I can see when they started to spike exactly.
And these are PCR tests, correct?
This is PCR tests, yes. So we started initially with the CDC's PCR test, and then we added capacity for some additional RT-PCR tests so that we could scale. We're currently running the Thermo Fisher TaqPath COVID-19 test in the lab. We also have capacity to run the Solgent Diaplex as well.
And at the same time, you're doing influenza surveillance, right?
Yes, we're still doing our influenza surveillance. We're still doing all of our enteric disease surveillance. We're still doing STI testing, syphilis and HIV, and tuberculosis testing.
Okay.
We rapidly had to scale from having, as a reference laboratory, we ran roughly 8 to 5 days a week. We had staff who are tasked with coming in if there's an emergency because we're part of the laboratory response network. So our staff were used to the idea of there might be a sample that has to come in, and we're going to ask you to come in Friday evening or Saturday to run a sample. But we rapidly had to scale from that sort of operational mentality to we're currently running 24/7, 3 shifts in the lab a day, 7 days a week.
Wow. So your lab does the PCR reactions, right?
Yes.
And you do all these pathogens that you've mentioned. Your lab is doing the screening. Where do the samples come from typically?
So for COVID-19?
Yeah.
So primarily in the beginning, we were the only laboratory in the state that was able to do testing. So we were getting stuff from everywhere, from local public health agencies, from hospitals, from community testing. But that's evolved, thankfully. And so we are primarily focused right now on running testing for facilities that are state-run, congregate care facilities. We do a lot for the Department of Human Services and the Department of Corrections. We also do some community testing sites and outbreaks in long-term care facilities, although the routine long-term care facility testing that's done every week over and over again is now moved to a commercial setting.
And have you added any antibody tests or other tests, or is it strictly still the PCRs?
So we have not added the antibody testing. We have not had a lot of requests for it. We had a plan in place and we're ready to go to get that out the door. But at this point, we haven't had a lot of demand. But I honestly see that potentially changing as we move to this vaccination world that we're heading into.
Emily, have you had any dealings with the Native American peoples in Colorado? There's a lot of settlements there, and they've had a lot of problems with this, obviously. Are they making use of your facilities? I hope they are.
So they are. They have access to their own testing, and the government the federal government level has sent testing supplies directly to some of their locations. Oh. But we also run rapid response teams here out of the laboratory that stand up community testing sites around the state, and we have held sites within their facilities as well, and then those samples come here for testing.
Right. Thank you.
Emily, there is a CDC in Fort Collins, right? A branch?
There is, yes.
And do you interact with them at all?
We do. We interact with them quite a bit. When this first happened, they reached out to us, and we met with them, and we talked about how they could help support us, and they did help getting us moving with some equipment, some expertise. They did help us with that sort of scale of expansion. We've moved now, though, to their focus is primarily on the arboviruses. And so they basically reached out to state labs and said, we know that you're focused on a lot of this COVID related things right now. Can we take some of your West Nile surveillance and other things on that way and sort of shift things around as to what's being done in each location so that we can meet the demands overall. But they are fantastic partners, and I'm really quite thankful to them because they were the ones who first brought us some of the automation. We didn't really have a lot of actually we had almost no automation for extraction at the start of the pandemic. And without their assistance, it would have taken us a lot longer to scale up.
So mainly what kind of samples are you looking at? Are they the nasopharyngeal swabs? Are they nasal wash? Are they saliva or all?
Yeah. So most of the samples we get now are the anterior nares sample. At the beginning of the pandemic, it was primarily the nasopharyngeal swab. But most of the community testing sites and the repeat testing, they found that people don't like to have the back of their brain tickled multiple times a week. And so while that does still does remain the gold standard sample, if you want to get compliance with having people who are living in congregate care settings tested on a regular basis with a PCR test, we found there's better adherence to the nasal sample.
Okay, so towards the end of December, I suppose, it was reported from your state and your laboratory, I presume, that the B117 variant was picked up. So can you tell us about how that came about?
Sure. Around the 20th of December, we had received news that the UK had a variant of concern and that it had mutations in the spike protein. By the 21st, we had raised our staff awareness on this and set up a screen that any sample that was run on our Thermo Fisher TACPath COVID-19 test that had a characteristic profile was flagged to be sequenced. So we are set up to do whole genome sequencing on COVID-19 samples, and we were already running that routinely in the lab. And so beginning right that week, we went to our freezers and started going through everything that had come up positive and looking at the actual run curves off the instrument to see if anything met that S dropout profile that is characteristic with this assay. So the COVID-19 TACPath assay has 3 targets, and they are the N gene, the ORF1AB gene, and then the S gene. So the mutations in the S gene happen to be at the same place where the primer probe combination sits down in this assay, and so it can function as a screen. And so we went back through those freezers and looked for everything we could find, and we had somewhere around 24 samples. And this was done by Dr. Shannon Matzinger in my laboratory. So she's our lead sequencing scientist, and she started to put all of these immediately onto the instrument. And many of them at that point had really high CT values overall for the sample. And we have noticed as a trend that the S is the first signal to disappear when you have really, really high CT values. And so we didn't see any sequences that were the variant from these samples. But then on the 26th of December, we had 2 samples come in that when they were initially run through that diagnostic RT-PCR test, had low CT values and no signal for S. So they had, one of the samples had signal around 12, 13 CT for the ORF1AB and the N gene, and the other one was in the low 20s for the 2 genes, but no signal for S. And so they were immediately put onto the whole genome sequencing. And on the morning of the 29th, at 6:00 in the morning, I received a chat through the Google chat that the state uses from Dr. Matzinger in the lab saying it's here. And I immediately picked up the phone and said, "What's here? What are you talking about?"
The TV people know.
Information.
Yes.
Right. So, at that point, it was, we went through and looked at the sequence and then shared it immediately to CDC. And they said, yes, looks good. Or
rather looks bad.
True. Exactly. Right. So the data looks good. The results maybe not so great for what comes next.
What kind of information did you have on these samples that might alert you to where they may have come from?
Yeah. So, it like everything in public health, this one was a bit of a mystery right from the start because they had come in with incomplete paperwork to our laboratory. Right. And it didn't, we had patient name and ID and no submitter name. And so we were looking at these results and immediately reached to our epidemiology partners within the health department and said, we need to dig into where do these come from. We have a fantastic team in our accessioning group, which is our data entry team, who was able to help us as well, sort of piece back, well, they came in at this time of day and, this is what else they came in with. And we looked at the names and we were able to determine that they belonged to the National Guard. And they were part of a team that had been deployed to assist in a long-term care facility that was having an ongoing outbreak. And they were providing some backup staffing support to the facility. And so we thought, oh boy, this could be really, really big, right? So the samples for that long-term care facility outbreak, though, they weren't coming here to the state laboratory. They were going to a commercial lab. And so we didn't know if we were dealing with 2 people that had the S dropout variant profile, or if we were dealing with an outbreak of that. And so we sent our rapid response teams out there and they tested everyone, and we ran diagnostic PCR testing on all the patients from the facility, and we did not see the S dropout there. So we had a bit more of a mystery at that point.
I want to back up for a minute because you said initially that you had 2 low CT value S dropout samples. Were both of them, are they different individuals, and did both of them sequence to be the variant of concern?
Yes. So they both are, they're two different individuals, and they both now have sequence confirmed to the variant of concern.
Okay.
Initially, though, when the state made the announcement, we had full complete sequence on the sample that had the lowest CT values, the sample that was 11, 12, 13 CTs. The other one, we were missing just a little piece of it. And all of the other spots across the entire genome looked great, but some of those key mutations, we just didn't have signal on them. And we wanted to ensure that we had the full sequence there before we called it the variant.
So do these two samples have the same last name?
No, they don't. They do not. Yeah.
Were they
But
symptomatic?
they were the same. Were they in the same National Guard unit that went to that facility?
They were, but they did not get it at the facility. So that piece we know.
Right.
That's very interesting.
Were they symptomatic?
One of them was symptomatic.
Wow. And what was there, any idea about their travel history for the?
They did not have travel history, neither of them.
I'll be darned.
Yeah.
And so when did they test positive?
They
December
tested
20.
positive on the 26th, but those samples were collected actually on the
After
24th. Yeah.
Merry Christmas.
Yes.
Exactly.
Exactly.
So you put these.
Sorry, Vincent.
If you put the sequences on the tree of all the other VOCs, can you get a sense of where they originated?
They're clustering with all of the variants going right back to the sequences that were coming out of the UK. Yeah. And they're looking like very little divergence from that.
And since then, so you're continuing to monitor for this, through the routine PCR, you're continuing to monitor for the loss of this fragment. Seems like it makes it fairly straightforward to continue the surveillance.
Yes.
Have you seen more of it?
So we have yesterday. We have a third one in Colorado.
Okay.
Yes, as of yesterday. Mm-hmm. And this one is not necessarily. They have not completed all of the epidemiology contact tracing on it, but it appears to be that an indication that we're looking at something that probably happened in the community.
Does this cause a radical rethinking of how this virus is transmitted?
I don't know about radical rethinking. I mean, I think it's out there, and it is. I mean,
what if somebody sent a package from, let's say, the UK, if I dare say so, to a relative in, let's say, Colorado, and it contained material which allowed the virus to survive long enough? I'm just speculating here because I know that some strange things happen that we are not expecting until we get wind of how these things are actually spread rather than the way we think they're spread. So have you speculated at all as to where these three might have, without traveling outside of Colorado and without knowing each other and without commingling, seems kind of interesting. You have a writing of a novel in this one, I think.
Well, I do think that this is going to tell us a very interesting story about how this virus is spreading. I think being able to identify a characteristic variant like this in the sea of all the COVID positive tests out there does give us this opportunity to really connect with the epidemiology side of the spread of the virus and look at where is it moving across the country. I personally think that we're going to see that there is some transmission of this within our community now that we've seen this third case. And I know there are other samples that they have not ruled in or out yet in the lab. They're too early for me to tell you counts on them yet. They just are, looking at that initial, there's at least one other S dropout that hasn't even moved to sequencing yet. And I don't honestly know if they've even looked at the CT values. That was just this morning. So it might be too soon for me to say that we have more than three, but I think I imagine that we are going to see a few more than three, and we're going to be able to look at networks of transmission and see the travel associated bringing this to Colorado.
So I've seen reports that this has shown up in a number of different countries. Mm-hmm. I've seen like 30 or 32 or something like that. I don't know how many or how well confirmed it is. I'm wondering if you can elaborate on that. And in my mind, that suggests to me that we really don't know where this came from. Okay. Is there any way we can have any idea of the real origin of this at this point?
I think at this point we need more sequence data worldwide to really answer what you're saying there, because it was detected first in the UK, but it's not necessarily true that it arose in the UK.
Right.
I mean, these viruses they're mutating all the time, and until we have either sequence data or a failure of our diagnostic test, we don't really know that it's there, right? So I do think that it is probably much more widespread than we know at this point.
And correct me if I'm wrong, but wasn't it, in going back to earlier samples, haven't they found that in the UK it was actually there as early as September or so?
Yes.
And then, but then it ballooned, possibly just as part of this big surge of cases that they were having in November, December.
Exactly. And I think that to me was very interesting, because that big balloon in the number of cases that were attributed to the variant seemed to correlate with when they were in a period of more social lockdown. And I think that does lead us really to have some concerns about transmissibility of this strain, that if one person has it, they may be able to infect more people than if they were to not have the variant.
Now, I actually the lockdown issue, my understanding was that different parts of the UK were in different levels of that at the time, and there was actually a fair amount open in some places and maybe not in others, and I don't know to what extent that might have played a role.
I mean, I think that the levels of lockdown in the various populations is playing a huge role in how we're seeing spread. Even in, here in the U.S., when we have different segments of our society,
Yes.
when we close the restaurants, we see different effects than when we close the schools, for sure.
Yeah.
So, Emily, how well connected are the state public health laboratories to each other so that you can have an instant read on who's seeing what, to know exactly how many cases there are of this throughout the entire United States?
Yeah, I would say that the state public health laboratories are rather well connected through the Association of Public Health Laboratories, APHL. We have a listserv where all the laboratory directors talk to each other pretty regularly, and we meet and discuss these things. And I would say that there's very good communication on these electronic routes, and we freely reach out to each other all the time. In addition to having the national group, we also have some regional networks.
Right.
So we're talking to the labs within our region probably even more frequently than labs across the whole country.
Yeah, so my question then is, knowing what you know and what everybody else knows at the state level, how many actual cases of this are there in the United States as we speak?
I would have to look in my email, because I think it's changing just that rapidly.
It changed 2 minutes ago,
I think it may just be changing that rapidly. Yes.
Wow. Were you the first state to? You weren't the first state to see this, though, were you? Or maybe
we were. Colorado was the first in the U.S.
Okay, so December 26th. From that point on, how many cases do you know about now?
I think we're up to in the 30s, but like I said, I'd check the exact numbers for today.
And there's a scatter diagram that shows where these are, and they're not related to anybody, and there's no sense yet, or
You're talking about
it
states,
looks like
Dixon?
a pattern.
You're talking about states? You're talking about
State
U.S.
levels.
states?
Yeah, statewide. Yeah, I want to know how many other
We
states.
got California, right? New York,
California, New York,
Florida.
Georgia.
Pennsylvania
Oh,
just happened,
Just happened.
okay.
or just was detected, I should say.
So Emily,
Thank you.
in this time since the 24th or so, you've got 3 of these B117. How many other SARS-CoV-2 infections have been confirmed?
How many other in Colorado infections have been confirmed?
Yeah, in the same period where you've picked up these 3.
I think we're seeing somewhere around. I should have pulled up the exact numbers.
Probably thousands, right?
We have. It's definitely in the thousands.
Okay.
We're seeing every day.
Yeah,
Yes.
because I think what's going to be very interesting, if in fact this variant is more transmissible, now that it's seeded in Colorado by whatever means, if the secondary attack rate increases, then we should see more and more of it. If you see sporadic cases here and there, and over the next 6 months, 20, 30 cases as opposed to 10,000 or 20,000, it would tell me that it's not particularly more transmissible, at least in Colorado, right?
Right. And we're doing a couple different things in Colorado to try to answer that question. One of them is that we have, of course, asked every other lab in the state that if they see this dropout, we want to have it come here so that we can sequence it. We've also stepped up our overall sequence surveillance across the state, where we're asking labs to send us a subset of their positives every single week so that we can sequence them without requiring them to have this S dropout, because I want to make sure that we're not focusing only on this one particular variant. And also not all platforms can detect that with the diagnostic test. And then really working closely with our epidemiology partners on the contact tracing on these cases so that we can any facility that they may have worked in or contacts that they may have had, particularly since we went over this holiday period where people may have gathered, to make sure that we then send our rapid response teams out to collect samples from any known contacts that are being given isolation orders of any kind.
And how long are you recommending that they remain in isolation before they can come back out?
So we're currently using a 14-day timeline for isolation for cases that have any contact with any suspected variants.
Right. So is that entire unit of the National Guard now in isolation because they've had contact with these two people?
It's not the entire unit, but those who were deployed to the same facility for that staffing assignment are. Yeah.
So
and what's the overall situation in Colorado in terms of restrictions? What's opened? What's closed?
So right now, Colorado has a dial system that uses sort of a color code metric to let you know how much social isolation you should be observing in your day-to-day. And as of right now, most of the state is in sort of a yellow-orange, which is a concern and high risk. We're limiting gathering sizes. Restaurants are open at this point in most parts of the state, with restricted numbers of people who can be inside. And the schools are all looking to work on trying to return students this week and next week to in-person classroom among the young students, and then more of a hybrid model as the students are older.
I'm looking
Do you have
at
an estimate as to how compliant they are?
Compliant for the
to the orange, yellow restrictions.
Yeah, overall, there's very, very good compliance. Just before Thanksgiving, many of the counties were moved to a red level, which was a higher level of control or isolation. And there was a lot of compliance with that. But now that we've come through that holiday season, things have moved more back to the orange level.
Now, in terms of compliance, one of the issues that's come up repeatedly is you can tell restaurants what to do, and it's easy to go around and check if the restaurants are letting too many people in and so forth. But it's a lot harder to see if people had, all 20 of their relatives come over for Thanksgiving or for the big Christmas, feast. So is there even any way to get a read on how?
Yes, there is actually. They look at movement of cell phone data.
Okay.
Yeah, and also traffic patterns as well.
Yes.
And so I think those two indicators have shown that, at least here in Colorado, that people are listening, and they are. I can't say that two people didn't go to grandma's house, but. There's not been as many, as many people on the roads, and that's been indicated by there hasn't been as many, DUIs even in Colorado. So
how about pre-Christmas flights? You're near an enormous airport. I've been to that airport many times. Mm-hmm. And how will Coloradans, with regards to, stay at home and have your holiday with a few friends, but please don't travel. We saw an enormous number of people just get on an airplane and go someplace.
It was a lot, I think. And
the airlines are not under that restriction because they're sort of immune to all of this because of their financial situation. I think. I'm not sure.
Well, no, there are restrictions, but they're in a
full flights
spot.
Are the typical.
And what we saw nationwide over the Thanksgiving period was a huge spike in travel, and then over the Christmas period, we actually saw the highest levels of airline traffic that we'd seen since March. So,
I want to.
Yeah, that's.
I wouldn't pick on
concerned.
Colorado for that.
Want to show the map of the U.S.
Oh,
here.
yeah, that's right.
You know,
That's a New York
Colorado
Times.
is not the reddest state, that's for sure.
Oh
No. Arizona now I think has the.
Yeah, but that's mostly Native American reservations. I think you're looking at there.
I think it's cities in Arizona.
So,
Yeah,
Emily,
but there's only one city in Arizona.
Yeah, but it's loaded with SARS-CoV-2 right now.
Yeah, but it should be a red dot right in the middle rather than what you're looking at, I think.
so Emily,
That
this
area down near California border,
Yes.
that's mostly Native American population.
there are a couple of counties in Colorado that seem to be, particularly heavy, but the rest, some of them are none recently reported. Right? Probably nobody lives there. I guess.
Well, no, that's the people, the original people live there. That's the point.
I'm talking
Not
about
always.
Colorado,
Right.
Dixon.
But
I know. I'm talking about Colorado. You see that very, very red dot? That's
Right here,
where Emily is. That's where where it says Col.
right up here.
Just to the left. That's where Emily is right now, and the rest of the state is pretty.
Well, let
less dense.
Emily tell us. Is this where?
that's Pitkin County. Actually, that's up in the high country, and we do see a lot of travel associated with that. That's actually one of the hot areas of the state that was hot initially in the pandemic as well. And we have a lot of ski resorts and people traveling from across the country that head to that part of the state. So we do make sure that we have a lot of community testing, and I know we're focusing some vaccination in the populations up there that are meeting the current CDC phasing guidelines to target where we have a lot of cases as well. And
in fact, over
county, sure.
Yeah, there was the FAA sent out an alert about air traffic congestion, which is the first time this has happened in quite a while. That there was severe air traffic congestion in snow country destinations, which would include the high country of Colorado and also South Florida over the Thanksgiving and Christmas breaks.
Emily, back to this the sequencing and the dropout. So I just looked over at the CDC site, and they said of the 17 million, and this is from the end of December, of the 17 million U.S. cases, only 51,000 have been sequenced. And you said not every PCR test will pick up the dropout. Is that right?
That's correct.
So we could have more than we know at this moment. We just haven't looked hard enough.
I think that's true. We were fortuitous here that we were running primarily on that Thermo Fisher TaqPath test that gave us the ability to see the fingerprint, but not every sample is doing that. And I know that the U.S. in general is moving towards more sequencing, and I'm really glad to see that happening. CDC has ramped up what they're doing across the country. We were submitting 10 samples to them on a routine schedule, and we've increased that number now to 24. I think it's every 2 weeks, 24 every 2 weeks to CDC on top of what we're doing here in the state. And we are, we've done over 1,000 genomes in Colorado, and trying to get to the point of being able to do close to 200 a week to just really get a better picture of what's going on here and contribute that into the national databases and get them into next strain so that we can see where things are moving.
How do you decide what samples to sequence?
So that's been interesting. Initially, we were looking at outbreaks or clusters. We sequenced some outbreaks that were associated with university transmission, with some of the fire camp transmission. We also were focusing on trying to look at whether or not we could use sequencing to answer questions around repeat infections, and got some interesting sequence data that showed that potentially we may have something that will turn out there. But I think we've really shifted hard towards this variant sequence right now. And I keep cautioning and trying to pull us back and make sure that we are getting this subset of samples across our state so that we can see everything that's going on. Because if this variant is here, I'm convinced we're going to find something else or other molecular signatures that we can use to look at relatedness in Colorado and how things are moving.
So,
and that difference in tests also raises an interesting point about why this was first seen in the UK, right? Because their standard, they had a very high standard for doing routine sequencing, and then they picked up this variant, which just happens to cause an easily spotted difference in the standard PCR test. So then you can cull out those samples. It's almost like this variant is especially well labeled for detection, right?
Exactly. And I think, from my lab's perspective, what happened in the UK really underscores why we need to have public health laboratories that are doing this sort of in between diagnostic testing. And it's not exactly a research project, because we're asking more population level questions around what's going on with the samples that are available within our state. But, if you're a person with symptoms, your most important question is, do I have COVID-19? And that's what a diagnostic test will tell you that. But then being able to take a subset of those samples, or as many as we can get our hands on here, and do the more deeper dive into the genomic sequence and ask what's happening with COVID-19 on a population level, instead of on an individual level, and try to take that and connect it back to the individual and where their contact tracing networks go and what is it showing us about transmission, it's been powerful. And I really look to see more of that coming out as we expand sequencing.
Once
Emily,
you get that data, you will know how to predict the future. You can actually see this evolve in front of you, just like we did for the West Nile virus. When the weather patterns change and the mosquitoes disperse and the birds fly away, you're going to have some human cases, and horses too. But this is different. This is, predicated on compliance to staying at home and all kinds of other anthropomorphic stuff that state control is usually not available for. So I think you're going to have an interesting book to write about this story.
So Emily, you mentioned getting some test information from some other facilities in Colorado. Is your lab the only lab that is doing sequencing, or are others capable of that sequencing?
So there are some other labs in Colorado that are capable of sequencing. Some of the large universities are also kind of pivoting in this direction. And I know that University of Colorado and Colorado State University have both have some active sequence programs where they are doing this. They have not found any of this specific variant yet. They are contributing their sequences into the national databases as well. And we're looking to have I'd like to be able to display our sequence data in a way where you can see sort of the Colorado profile and then overlay over the national profile, so that we can really pick that apart.
Cool. Now that you've seen this variant in your lab, has that changed the way that your lab is sort of conducting any aspect of the testing, or do you see that changing any aspects of public health guidance?
So it has highlighted the need to really keep the message strong about we need to be wearing our masks and doing our social distancing, particularly with the concern that the variant could be more transmissible. And I think that the time in which we found it, I'm hopeful that getting that message out right before the New Year's holiday hopefully influenced people to make good choices over that New Year's holiday. So that for me was one of my motivating factors in wanting to really talk about this variant publicly and make sure that people in Colorado were aware that it was here and what that risk could mean to them personally. And then in terms of what are we doing in the lab right now, every analyst, every technician, every scientist in the lab is aware of what the profile looks like. And they're all involved in trying to help search for the next one. And so I think it has also helped us with our just our staff engagement in the project. I mean, this pandemic has been going on for a very long time, and having all of the staff know that their work has led to something that they work the third shift at the state lab overnight over working on Christmas or whatnot is not necessarily the most desirable job that they might want to be having, and to know that the work that they're doing has resulted in this thing has been very helpful as well in just keeping the mission moving forward.
Emily, am I correct in assuming that the public health department has some epidemiologists or a team of epidemiologists on board? And if so, I'm curious as to what their focus is during the pandemic, and maybe even specifically with respect to these variants?
Yeah, so we have a very large team that's also ramped up, just like we had to ramp up testing. Our epidemiology group also had to ramp up the number of contact tracers and trained epidemiologists they have on staff. They are primarily focused on tracing down and controlling outbreaks in really critical populations in the state of Colorado to make sure that we are controlling outbreaks in our prisons and our long-term care facilities, as well as community spread outbreaks. And there is a specific team now that has been dedicated to the variant, to trying to ensure that they are not spread. It's not spread beyond those initial identified cases.
So the epidemiology is integrated with the molecular diagnostic efforts as well. So
it is. We work very closely with them, and we even added a new meeting now every morning to touch base with the epidemiology and the lab, specifically on the variant cases.
Okay.
Emily, do you have any involvement with the vaccination efforts in the state?
So we do have some involvement. We have these rapid response teams that were stood up to support testing around the state, and we are now taking sort of that infrastructure to support distribution of vaccine as well. The state laboratory, because of its centralized location and the fact that we have backup power and ultra cold refrigeration capacity and freezers, made us an ideal choice to be one of the hub locations for vaccine to come in through the state. And, we did have some come here, get delivered. The first dose that came in, our governor actually came and received it from FedEx at the loading dock, which felt like a turning point, I think, in the pandemic response, just that, the vaccine is finally here, and we're going to use the resources, the logistics teams that we'd put in place to distribute testing supplies initially, and then rapid tests, as well as now use those same courier mechanisms and whatnot to get the vaccine out there as well. So we're just one of many locations in Colorado that are doing that, but we have had part in that, and it's been kind of
so
exciting.
That's interesting. I'm interested. The press, at least, one of the hot stories has been difficulty with distributing vaccine or actually getting people vaccinated. And I'm interested in your perspective on that.
Yeah, I think, rapidly standing up testing was, we thought that was difficult at the time, right? But I'm seeing that vaccine is much more complicated because you have this, it's a 2-dose vaccine. So you have the logistics around getting your first one and then the second dose. And then there's also the layer on top of that of the people who are qualified to actually perform the injections. And so it's a completely different staffing issue. Whereas, when people are collecting their own nasal swab, you don't need the same level of trained staff as you do for someone to give a vaccine. So those issues have been new challenges that have presented with vaccination.
And Colorado I live in a small state where we can drive across the state in 2 hours. It's not really a major logistical challenge, but you have serious logistical challenges in Colorado, right?
We do. We're a very large state. Most of our population is located in the Front Range around the Denver metro area, north and south from Denver. And then in the middle of our state is a mountain range. So during winter, the passes will close, and we have issues transporting things from one end of the state to the other end of the state. I mentioned earlier that the state laboratory runs the newborn screening program, and babies are born all across Colorado. And those samples are very timely that have to get to the state. Some of the newborn conditions, if you don't get a result back within hours to early few days, can lead to loss of life. And so we had a pretty strong courier system that runs through the state of Colorado. And we have basically used the knowledge that we've gained from the years of running that courier system with our partners on that to help support how do we get the vaccine out? First it was how do we get testing swabs out to remote places in Colorado, and then how do we get those samples back in? And then now the new question is how do we get vaccine out there, and how do we do that while respecting the cold chain requirements as well?
I've been thinking some about this issue of getting second immunizations out, and in Texas, they have an immunization database that's a statewide thing that I don't know how the data fields in that are populated, whether, no matter where you get a vaccine, whether it goes into the database. And I don't know the extent to which that might be able to be used to figure out who needs a booster when and notify them. But I'm wondering if there's any such thing in Colorado, whether you have any insights into the utility of a facility like that.
Yes, Colorado also has a statewide system, CIS, the Colorado Immunization System, and all of the data, no matter where you have a vaccine, ends up in that same database, and it also is capable of pushing out alerts. And when you get your COVID vaccine in Colorado, you have to provide an email address and phone number contact, and so they will be sending alerts through the system and through providers as well. You have to tell them who your medical provider is. So they can make those connections and get that out there.
Okay, good.
Yeah.
And that's integrated with, say, the VA and the tribal governments and everything?
It is, yes. And there are health information exchanges in Colorado as well that these data can flow through into the hospital, EMR, medical records, et cetera.
Emily, what has the flu season been like so far in Colorado?
It has been quiet. It has been strangely quiet. You know, we thought we would be doing lots of testing for flu and COVID in the same samples. We spent a lot of time kind of preparing for the possibility of there being outbreaks of respiratory disease in a long-term care facility that we had to trace the outbreak back to its origin, and none of that has happened. It's all been COVID. We have not seen the same kind of flu that we normally do.
Is there any other winter respiratory illness that you track that's also gone down?
Yes. I think we were all expecting that enterovirus was going to be high this season, and we have not seen that one either.
Yeah, you were from the state that was among the first to see the D68 outbreak in 2014, right? So
yes.
the D68 was expected to peak, and we didn't see that, right?
Right, yeah.
Do you track rhinoviruses?
Maybe it'll be on odd-numbered years now.
Do you track rhinoviruses as well in Colorado?
We don't test rhinoviruses directly here at the state laboratory, but our clinical partners are also reporting that it's low.
Are there any sort of interesting changes in any other particular infectious diseases, maybe the non-winter respiratory diseases? Are people getting more food poisoning because they don't know how to cook at home or something like that?
We have had some very interesting clusters of foodborne illness that probably were due to things happening in the home like that. A few that we tracked down, and we did put out some messaging around with the health department around how to properly can your foods and how to observe good refrigerator storage temperatures, because it is an issue. We've all changed our lives so very drastically in response to this pandemic, and it's impacted everything. We've also seen major drop-offs in sample submission for sexually transmitted diseases, and so we have not seen huge change to the positivity rates, but we've seen big numbers of the overall number of samples coming in. So it makes us wonder what's really happening out there if we're not seeing the samples, and is there going to be a surge? At this point, we just don't know.
So, Emily, speaking of plagues, how's plague doing?
So plague, that's one of the things that we provide surveillance for here in Colorado, and it's endemic. We see plague periodically. It's not so much at this time of year, but we do see it seasonally, and get some prairie dogs into the state laboratory to be tested periodically, and some fleas.
Do you find a lot of people that are normally gregarious and want to go to restaurants and bars and have parties, et cetera, instead take advantage of the great outdoors and go out into Colorado proper to hunt a little more, fish a little more, maybe become exposed more to tick-borne infections and to flea-borne infections that,
Yeah.
I don't know. Was that happening this year or not?
Well, I can say the behavior piece has happened. The infections has not necessarily followed, but the behavior certainly has. We've had a lot more use of all of our state parks and our hiking trails, and people really trying to take advantage of, if I can't go anywhere, I guess I'll go for a hike. And
exactly.
Colorado has the weather for it. So
we should feel sorry for the trout, not for the people.
That's right. Yeah.
I wonder if prairie dogs are infectable with SARS-CoV-2.
I think Tony should be the one to know.
Right.
Yeah, I mean,
We'll
certainly there are a lot of prairie dogs in Colorado, right?
There are.
That's where the plague is coming from. Wow, interesting.
Right. Mm-hmm. Have any rodents
squirrels
been susceptible?
and squirrels. Don't forget the squirrels.
What'd you say, Alan?
Have any rodents been susceptible?
Yeah, deer mice.
Oh, deer mice. Okay.
That was Tony's show, right? Mm-hmm. And I don't think he got out to do any trapping this season, but he said in the spring he's going to go out there and look in the wild to see, because in the lab, they're very infectable, right?
Okay.
Well, we'll see.
Emily, it sounds like you made a great career move. This sounds fascinating.
Beautifully timed.
I do feel that the silver lining to this whole pandemic is that I was in the right place at the right time to take advantage of the training background that I had and the skill set, and hopefully I'm using that to do something good for the state of Colorado and public health.
It's a good thing you'd been there for 7 or 8 years when it hit. It would be tough to arrive
come in on your first date of this.
Indeed.
So, Emily, is your ultimate target the head of CDC?
I don't know if I would want to do quite that. I'm very happy here in Colorado. Yeah.
Okay. Well, thank you so much for joining us. Scientific Director, Colorado Department of Public Health and Environment, Emily Travanty. Thanks so much.
Thank you.
Bye-bye.
Thanks, Emily.
Thanks,
Great to
Emily.
have
This
you.
was
Thank
great.
Pleasure. This was a lot of fun.
We had a whole slew of emails about some things we said on TWiV 703, so I want to clear them up.
Yes, I also had a whole slew of emails and messages myself.
So these were some statements. We made two statements. One, that the UK is only using one dose of AstraZeneca vaccine. That's not correct, right, Brianne?
Right. So I think that I said that they were not doing the second dose at the 3 or 4 week time point. And that came out as they're not doing the second dose. In fact, they are doing the second dose at a later time point at 12 weeks.
Okay. Yeah, I mean, many emails I got, and people were very upset, but that's fine. Good that you're listening. And second was, Rich had asked Brianne to talk about about the second dose, the boost, and for the Pfizer Moderna, what governs the timing. And you had mentioned that it has to do with waning of immunity. So why don't you explain what you meant and what's correct?
So first of all, I apologize to everyone who for being unclear about the second boost for the second dose of the vaccines in the UK. And I thank everybody for pointing that out. And as far as the timing of the second dose and a boost in general with a vaccine, sort of the older way of thinking about immunology smushed the idea of cells becoming effectors and having an effector response and cells becoming memory type cells. People generally push those two things together when really they're two separate issues. And I sort of smushed them together a little bit when I was talking. The timing of the boost is related to when cells have differentiated or changed to turn into memory type cells so that they can receive that second dose. They can receive that boost when they are memory type cells and get the experience as memory cells. It is entirely possible that there are still some effector responses, especially maybe some IgG present at that time. It's about whether or not the cells have differentiated into memory cells and can receive the boost as a memory cell.
So the 4 week, which is being done for one of the mRNA vaccines for the boost, you could actually go 12 as we see for the AstraZeneca, but they just wanted to accelerate the whole trial, right? Basically.
Exactly. They wanted to accelerate the whole trial so that we could have an approved vaccine quickly and so that we can get people protected quickly.
Because I know that I get a boost for tetanus every 10 years.
Right.
But if they wait,
and you want the schedule to be at a pace where not only is the trial relatively quick, but also the vaccination schedule when people get it. I mean, if you get a shot and then you're going to go get a shot 6 months later, are you going to remember? And you're not going to be fully protected during that time. And so you do want to have those relatively close.
Guilty as charged. The Shingrix vaccine tells you to boost between 2 and 6 months after the prime. And I forgot about it, so I didn't get my boost until after 6 months.
Right. Well, the memory that I always have when thinking about this is a day in one of our very large lab meetings in graduate school where we were working on vaccine development. So you have a whole room full of people who think largely about vaccine development. And my advisor asked everyone to raise their hand if they had received the first dose of the hepatitis B vaccine. And then if they had received the second dose, and then if they had received the third dose. And it was quite striking to see the drop off in who had received all 3 doses, even in this room full of people who spend their time thinking about vaccines.
Well, that's one of the reasons that this concept of an immunization database interests me. And now actually nobody, even though such a database exists in Texas, nobody contacted me and told me that either that or it went to junk mail, told me that it was time for my boost.
Yeah.
But it could be used in that capacity, it seems to me.
Yeah.
It does. And I also was sort of interested to hear about that immunization database because to my knowledge, we don't have such a thing in New Jersey, at least not overall. However, there was a website that was circulated earlier this week where people are able to register for a COVID vaccine and put in details about things like pre-existing conditions and where they work and things like that. And it will tell them which phase they're in. And supposedly we will get texts and emails when we are eligible, when we're ready to be vaccinated or when we can be vaccinated. I know I was excited to sign up for that earlier this week, but that was, to me, was the first time anything like this had been at all possible.
There is a New Jersey immunization information system. And I just looked this up. Most states have some kind of a registry, or at least on paper, they have some kind of a registry, and at least people who get treated through certain health systems or major health systems that are integrated with that and will get their vaccination information into it. But there's variation across the map. I mean, it's the typical 50 states thing, right? You've got some states that are really sharp with it and it's a really well integrated system. And then some states, they've yanked the funding of it for paying for other things and it's operating on a shoestring. And so I don't know. It sounds like New Jersey's system is not exactly maybe a well-oiled machine at the moment.
Well,
but
what it makes me wonder about is whether that system works differently for childhood vaccines for people born in New Jersey and maybe doesn't work as much for someone like me who moved to New Jersey as an adult.
That's another big topic. And I've covered conferences full of people who talk about this sort of thing and try to fix it. Pediatric vaccines are universally much better managed than adult vaccines because kids are generally easier to keep track of. Though there is a problem where usually firstborns are much more likely to be right on schedule with every vaccine, and second-born kids are probably going to show up for the first doses, and you might see them again for the boosters. And it's the Kodak effect, right? You know, everybody's got lots of pictures of their first kid.
Yep.
So there are issues like that, but adult vaccines, people move around, and then when you've got 50 different state systems, I mean, I've lived in several different states. Most people have lived in several different states. I'm sure the state of Maryland has no idea whether I've been vaccinated against anything I've gotten in adulthood.
And do you know if the state of Maryland gave your information about past vaccines to the state of Massachusetts?
No,
of course not.
They would have no reason to do that. I don't think there's any facility for doing that. Right. Even certainly not for my generation.
I can go to my healthcare site and print out my kid's vaccination history, and for me, it's whatever I got last year, and that's
right.
It, right? No record. But this New Jersey site that Brianne mentioned, it quickly got overwhelmed. I actually got on and registered, and then they said, no, don't do it unless you're tier 1A, you know, and I don't know if it's still overwhelmed. Do you know, Brianne?
I don't know. I filled it out earlier this week. I actually filled it out right before it got totally overwhelmed. So I did get a confirmation email only 12 hours later to tell me that I was in group 1C. But a lot of people I've heard of who did it after I did have never heard
overwhelmed.
That.
Overwhelmed is better than underwhelmed.
Yeah, no, it's a good sign.
Yes.
This whole issue with reminding you of a booster. I mean, you would think you could deal with this with our technology, but I get email reminders all the time and I ignore them. You have to reset your password in 3 months. Okay, goodbye.
Exactly.
You have to reset it in 2 days. I don't reset it until it's too late. And so it would be the same.
Right.
Obviously, the thing to do is make a one-shot vaccine. If you can.
Yes.
Exactly right.
Unfortunately, most of them are not, right? So
Right.
Yeah, and there's the other problem that I've got. I live in New Jersey, but my doctor lives in New York. So if I'm going to call my physician and say, "Can you tell me when I'm eligible to come in and get my vaccination?" He can't tell me because I live in a different state.
You may be better off going to your local Walgreens to get it. I don't know.
Well, I'm waiting for the CVS around the corner to announce that the senior citizens are now eligible.
Right.
That's when I'm going to go. I think I'll just leave it like that. I don't think it's going to work any other way. Plus, there's all this extra vaccine sitting around that nobody's using, and if they don't use it soon, it's just going to go bad, right? There'll be some meltdown,
We're
and
doing,
I mean that literally.
Dixon, we're doing a good job immunizing the freezers.
We are.
To
Well, that's
steal.
apparently that's slated to change. The incoming
so.
administration is planning to, instead of holding back doses to deliver the second dose,
Right.
they're going to go ahead and ship what
it
they've
all
got.
out. That's
And-
right.
Yeah.
That's right.
Because
One
it's
would
still
hope.
it's being produced on an ongoing basis, and so you'll be able to do the second dose on an
Yeah.
ongoing basis.
Yeah.
Yeah, that's right. That's right.
All right, I want to do a paper, a preprint, a bioarchive preprint today, which I think is probably one of the more important papers of the year, actually.
Not only that, but it's just absolutely brilliant. I was so excited with this paper.
It's really
so-
brilliantly done, brilliantly written, and man, I think the implications are substantial. It changes the way I look at coronaviruses and immunity. And it's called A Human Coronavirus Evolves Antigenically to Escape Antibody Immunity by Iguia, Crawford, Stevens, Ayers, Klenhofer, Milvolt, Greninger, England, Book, and Jesse Bloom out of Jesse Bloom's lab at the University of Washington and the Fred Hutch. So,
And we have to give the usual caution that this is a preprint.
Yeah.
But it seems like the quality level of it is such that it ought to be peer reviewed.
I have
pretty soon.
subjected it to my own peer review, and I would say it's it looks pretty good.
And you're even reviewer 3.
Yeah.
A pain in the neck, yeah. So one of the big questions about SARS-CoV-2, how long does immunity protect us? And so far it looks pretty good, right? There's Shane Crotty's work that we've discussed and others that say it looks like it could be years. But then we have this elephant in the room, which we have talked about from day one, and Ralph Barrick was the first to mention it way back in February. Human common cold coronaviruses, we keep getting infected, right? Every few years, 2 to 3 years. And we have talked of a number of papers that document that. So that has raised this idea that first the idea, the immune response must not be durable. But then we have seen studies where, there's immunity there, but people still get reinfected. So it's not like the immunity to these common cold coronas goes away. It's there, but somehow we get reinfected. And so the idea here is, hey, do these viruses do antigenic evolution like influenza viruses? And I got to say, I've been saying, no evidence for that. I just blew it away because nobody's done the experiments to look at it. That's the problem.
That which is asserted without evidence can be dismissed without evidence.
Yeah, so
Yes.
I am guilty of saying it ain't happening because there's no evidence, but actually no one really looked. And they say the assumption of minimal coronavirus antigenic evolution is not supported by limited evidence to date. So there's very little evidence, but they take a really close look at it. And man, I got to hand it to them. This is just beautiful. So the question is, do human coronavirus, they focus on one, 229E, which infects people frequently. And they've got some old sera, and they looked polyclonal sera from people. Oh, it's so cool. Right. So
just open up the freezer and pull out those old serum samples. Yep.
So they focus on the spike in 229E, and they take a range of 229E isolates. So we have a bunch and this virus has been circulating for over 50 years. So we have a number of sequences. And the first thing they do is really cool. They do a phylogenetic tree of 229E spike sequences, right? And they say a couple of features are really important. First, the tree is clock-like with sequence divergence proportional to when the virus was isolated. Second, the tree is ladder-like with short branches off of a trunk. And this is a signature of viruses like influenza where immune pressure drives the population by selecting for variants. Third, the sequence is grouped by date rather than by the country of isolation. And this indicates typically, they say, fast global transmission. And so the date of isolation is a good proxy for the evolutionary position. And this virus evolves along a single trajectory in time. So they took 5 spikes and these are spike proteins from 229E viruses at 8-year intervals from 1984 to 2016. They made the genes for all of them. They made lentivirus pseudotyped viruses, so a lentivirus with the spike from SARS-CoV-2. They show they get viruses, they're infectious, they reproduce in cells, and then they say, let's throw human sera at them. So they can do this in a BSL-2 lab. There's no SARS-CoV-2 itself. And they have sera collected from adults between 1985 and 1990. I don't know where they got those. It's not that long ago.
So the question is, if your serum was immune in 1990 and you get the 2000 strain or whatever of or circulating variant of the same coronavirus, are you still immune?
Right. And they say that the sera came from the Infectious Disease Sciences specimen repository at the Fred Hutch, which sounds like an awesome repository that probably has lots of cool stuff.
Yeah.
Good. That's a good use of these archives. Excellent. It's like, museums are great archives for animals' genomes. All right. So nearly all the sera they got from 85 to 90 had at least some neutralizing activity against their pseudotypes with the 1984 spike. All right. And all the sera that neutralized these pseudotypes with the 84 spike had reduced titers against more recent spikes. And in some cases, there's no neutralization. In one serum from a 28-year-old collected in 1990, it neutralized the 84 spike at 1 to 125 dilution, but did not neutralize the 92 spike at the limit of detection of their assay. And they have another example.
I want to make sure that this is clear. A lot of this has to do with when the serum sample was taken. Okay. And we'll get to this in the next figure, but sera that were taken earlier do not neutralize viruses that were circulating later.
Right. And so I think the key point to the data that Vincent just mentioned was that the virus was from 84. The serum was from 85. So the person could have been exposed to that virus. And then when they looked again with a virus from 90, the antibodies didn't work. And so that antibody or that virus was not present when the antibodies were made.
And I want to make a correction. I overstated that. I say do not neutralized. We're talking about a variety of different quantitative
have consistently reduced neutralization, but inevitably do not neutralize as well.
Now, some of them do not, actually. Some of them do not.
Some of them
do not.
Yeah.
But this is not a matter of what an immunologist might hear from some of this is, oh my gosh, so you only neutralize against the first one you've ever seen. And this is not about that. This is in no way about that kind of thing. Yeah. This is about can you neutralize a virus that came after that you've never seen before,
came after
which
when
implies
the serum
that the
was
virus
taken.
has changed.
Right.
So what's the shortest interval between the time you can react to the virus and then now you can't? The shortest interval.
So 84 to 90?
That's a long time.
6 years?
Six to 8 years.
If you got immunized every year, that's not bad.
No, that's...
and effectively with 229E, people are getting immunized every few years, most people.
Yeah, we get infected every 2 to 3 years. So, I mean, it's a continuum, Dixon, right? It doesn't,
I understand
it doesn't turn off at 8 years. It slowly changes.
But it's relevant to our discussion about how long will this vaccine last.
Yes.
Well,
they're talking about intervals in their introduction or their abstract, talking about intervals of, I think, at least 8 years-ish.
Yeah.
I mean, if I had to get immunized against SARS-CoV-2 every second year, that's not a bad deal.
No, it wouldn't be. But we need to know. I mean, right now, we're all thinking it's not going to be an issue, but as we'll see, it could be from
but they could change the vaccine instantly because it's an RNA virus, right?
So, right. I mean, we can. So
that's not a problem.
Since we're going into implications here, you can.
Why not?
I thought we were going to do that at the end, but
I mean, there's more data. I'm sorry.
Dixon always jumps the gun. It's okay.
That's
no, because I'm not a virologist. Remember, I have these other questions.
Is so quick, it's rushing ahead to the next.
Thank you, Alan. Thank you, Alan. No, I'll shut up. I'm sorry.
No, you don't have to shut up. It's fine.
No.
But eight years, Alan. So the serum collected in this one case from a 28-year-old in 1990 neutralized the 84 spike at 1 to 125, but did not neutralize 92, 2 years later.
Right.
No neutralization. So,
right. So it was falling, and
boom.
then it's gone.
My
But that
sense
also
is
might-
that it's
that also might indicate that there was a change in the virus
Yes.
by 1992.
yes.
my sense is that this is not happening on the same as rapidly or on the same scale as flu. Is
Yeah,
that
I
a
think
fair
that's
statement?
correct.
Right.
I think they feel the same thing, although I think they don't really have enough data, they conclude.
Right.
Because as they say, hey, it turns out nobody's really checked this with influenza virus.
Yeah, the important thing. Yeah. Oh, good point. Yes, nobody's really checked it with human serum with influenza.
With
That's
influenza.
right.
With neutralization. They have checked
Yeah.
it with hemagglutinin inhibition, right? Because that's very easy to do, and they've done it in ferrets, but yes.
Well, and with flu, you have an additional complexity. So what we're looking at here would be what the flu people would call genetic drift, right?
Yes.
You got
yes.
changes in one protein, whereas with flu, you have that, and then you also have shift, where a whole new H and N combination comes in, and that's very different from what came before. And I don't think that happens with coronaviruses in the same way, because it's a different type of receptor, right?
Yeah.
They're a different type of antigen.
But the important thing about this paper is that this is the first real demonstration that this happens in coronaviruses
This
at
happens
all.
in coronaviruses,
Yeah.
yes.
I mean, it's human serum, and it's human viruses. So
It's the right experiment.
experiment to do.
Yes.
All right, so then they repeat this with another batch of sera, because they want to say, and another spike, because maybe this is a weird spike, this 84 spike. So they have sera from 92 to 95, and they look at neutralization of the 92 spike, and 18 of 19 sera neutralized this 90 from that range, neutralized the 92 spike-bearing viruses. They neutralized the more potent sera, neutralized the older 84 spike again, but had lower activity against spikes from viruses isolated after the sera were collected, after 1995. So they say, looks like the 229E spike is evolving antigenically. So immunity made by infection with prior viruses is ineffective or less effective at neutralizing future viruses. Brand new discovery, really. I think really important. I think it changes the way we think, because we always say, they don't antigenically evolve. They do. So they've tried to quantify the rate of evolution. Like we were talking about this a little bit as well. And they say that the neutralization by some sera is eroded over tenfold by just 8 to 9 years of viral evolution, whereas neutralization by a few sera is unaffected, even by 16 to 17 years of evolution. So it's not the same in everybody, clearly. And, okay, so this...
But again, just to be clear, we're not getting into whether the antibodies are waning so much here. We're getting into whether the antigen changed.
Right.
Yes, right.
So that's the
Right.
question
And
next.
in fact, the way this experiment is done, it is explicitly not looking at antibody waning, because they're using the same concentration of antibodies.
Yeah.
So we assume everybody's just generating just as much antibody in 1990 as they were in 1984. You throw the same dose of that antibody at the 1990 virus, and it's not effective.
Yeah. Now, the next question related to what Brianne said, if you make antibody to a virus in 1984, do you maintain a long time the capacity to neutralize, or does it wane, right? Does immunity durably retain the capacity to neutralize historical strains that you were exposed to many years ago? That's a different question, right?
Different question. Requires a different experiment.
So they have modern sera collected in 2020 from children and adults. The modern adult sera that potently neutralized the 2016 spike also neutralized all the other spikes back to 1984.
That is if it's collected from an adult.
Yes.
Adult sera.
That's what the brilliance of this experiment.
Yes.
I love it.
So if it's somebody who lived through 1984, they can still neutralize the 1984 virus. And if you weren't there, then...
Yeah, so from children's sera neutralized the spikes that were circulating in their lifetime.
Yes.
but not before they were alive because they couldn't be infected with those other isolates. Right.
So for all of you who are planning time travel,
You better get
Alan.
vaccinated.
you may have to allow for catching a cold when you get to your destination, depending on which direction you're traveling and whether you've been there before.
It's obvious that we're still reliving 1984 in other ways.
Yes.
Okay, so they say this further emphasizes that the reduced neutralization of future viruses is because of antigenic evolution, not the waning of immunity.
Right, because there are ways that you could think about some immunological explanations here. And I think they do a really nice job of teasing apart is this the antigen or is this an antibody change? And so we should be clear that this is really about antigen change.
So a little bit of a backwater here, and I'm sort of looking at you, Brianne. I'm thinking about, because you were referring, I think, earlier to the concept of original antigenic sin.
I was.
Okay. So I would think that if that were going on here, let me see if I've got this right. If that were going on here, then older people, their prevalent antigenic specificity might be the older viruses rather than the newer viruses. Okay, but I'm not seeing that here.
Yeah, I agree with you on both parts of that is sort of the way people sometimes think about original antigenic sin, but that is not what they've seen here. We need a lot more information to fully be able to make much of any conclusion. What that might tell us is that usually when I explain original antigenic sin, I talk about how the old and the new virus have some epitopes that are different and some in common. That are part of the process. And here I might imagine that these two viruses have nothing in common epitope wise. And so original antigenic sin is less of an issue. But
So
again, I need a lot more going on.
Okay, so in order to really look at this further, you're going to have to drill down to individual epitopes and the whole spectrum of the antibody response in given individuals.
I would think so.
At the last part of this paper, they say, where are these changes in the spike occurring? And they look at the sequence, of course, and the spike, of course, is divided for our purposes into S1 and S2 subunits. And, of course, the S1 subunit has the receptor binding domain, for example. And they see most of the sequence variability among these different spikes that they have studied in this paper is in the S1 subunit, particularly focused in the receptor binding domain. So what they do is an experiment where they swap out the receptor binding domain of the 1984 spike with the RBDs of all the other spikes that they've used. So they make chimeric spike lentiviruses, basically, and then they do their neutralization assay. And they can show that the drops in titer are paralleled by drops in titer against the RBD. So the major changes are in the RBD, but not every sera showed that. Some of them didn't show a correlation with the RBD. So they think that, there are other parts of spike that are probably varying as well, not just in the RBD that are explaining this. And they obviously could have done a lot more here, and they probably are with single amino acids and so forth, but they're saving that for another paper.
Right. I think that one could, make a career in immunology probably based on trying to figure out the exact details of what's going on with all of that and the epitopes and how they're impacting one another.
I'm not sure about a career, Brian, but you could get a couple of papers out of it.
You could get a career started with this.
You could certainly get a career started with it. I'm just wondering if there are interesting immunology mechanisms behind it that would bulk up more at the career.
Yeah.
So for the audience, I'd like a little bit of discussion of some viruses. So we're talking about antigenic variation here. Okay. Some viruses do this in a big way, like flu. Some viruses don't, like measles. Mm-hmm. Why?
Well, measles and polio, they can't, well, as they say in their discussion, they cannot tolerate those amino acid changes that are needed to escape neutralization.
Right.
Those that can do it do.
So
Right.
I think is probably the answer.
Influenza virus, HIV, the C229E, common cold corona, they can all do it. All these viruses have a relatively high mutation rate. And as they say, SARS coronavirus is a bit lower because of their error correction. But nevertheless, every position that can change, yet it in measles and polio, you never see it. And probably it's because they're constrained, right?
Right.
Yeah,
So
exactly. So it's a structural constraint. The part of the virus that would need to change in order to avoid an antibody response, changing that part also destroys the structure of the virus and makes it not work anymore.
Now, do you folks remember a long time ago on TWiV, we did a paper, and Alan, it's like how they used to build airplanes. They would shoot bullets and see what parts they had to reinforce.
Right. Yes.
Remember?
Which the bombers that came back, you look at where the bullet holes were in the bombers, and then you reinforce the areas that weren't hit.
Okay. So what does it have to do with viruses? So they took measles and they shot the entire hemagglutinin protein, one of the spike or whatever it is that's conserved. And they found regions that could not change, and those are the epitopes.
Because the plane doesn't come back if those places get hit.
Right. And I think our title reflected something about that, but I don't remember.
Yeah, I think that there's a famous diagram of this that shows, I think, a B-17 in profile and little dots all over it. And there are a couple of places like right around the cockpit,
right under the
wings, where you don't see any dots because those planes didn't come back. And those are the places that needed to be reinforced.
So it's not a why question, because that's an anthropomorphic thing. That's where the PH for the PhD comes in. It's the data. It's the how thing. It's a how. How do viruses escape? And they escape because the non-essential parts to the virus's biology are changing, but they're very important in terms of the way our immune system visualizes it.
If our immune response is against a part of the virus that the virus can afford to change and still be able to infect, then the virus will evolve around that. And if our immune response or some other aspect of our biology is around, like the poliovirus receptor, is around a part of the virus that can't change in order to do that function, then...
No, Alan, you're contradicting yourself. They do change, but when they do, they don't come back.
They don't come back, exactly.
So they do change.
Yes,
they change and they don't succeed.
For them, it's a lethal mutation, but it's for them, not us. So the point is that mutations are random.
Yeah,
of course.
Totally. So the ones that are random that don't matter, that's one explanation. The ones that change that do matter, that's it.
And
so remember, we are a natural population of animals. We are subjected to all the pressures of evolution as every other animal is. And so therefore, our immune system evolves along with that concept, and you are what you are.
Except that we've actually broken the game. We're not subjected to all the selective pressures of other. We are a selective pressure.
It speaks for yourself, because if you live in sub-Saharan Africa, you still do. If you live in places where you have no medical interventions,
as a species, we are more a threat to other species than they are to us.
Oh, no, there's no question about that. That's true.
So so in terms of implications for the vaccine, which was where Dixon was trying to go earlier, and we forced him to slow down and grind along with us through the paper. I think you could see two ways that this could go, right? You could say, well, either we're going to have to generate a new vaccine every few years when SARS-CoV-2 mutates its spike to the point that it evades our immunity, or it won't matter. Because if we look at the previous coronaviruses, we've got 4 common cold coronaviruses circulating in humans. At some point in the past, and we've talked about this, they spilled over from animals.
Yeah.
And when that happened, it might have been a global pandemic, just like we're going through now, but nobody noticed because people had such a short life expectancy and they were dying of so many other things that some more people dying of bad respiratory illness was just like, oh, wow, that was a bad year. And now we see it happening and we can actually appreciate the scale at which that's happening. And so we're developing a vaccine, and we get these vaccines and we're going to vaccinate everybody, hopefully. And then we'll have immunity to the 2020 version of SARS-CoV-2. which then continues circulating and evolves and gets around that immunity over a period of 3 or 4 years, but everybody's catching it again next year. And maybe it doesn't, maybe it's not severe when you catch it again. Like maybe it's like 229E or OC43, where you catch it, you get a little sniffle, you get over it, even if you're in what we're now calling high risk groups, because they weren't dying of 229E, right? So this could just be a sort of an endogenization of the virus, and the vaccine is going to ease that process.
So didn't all the people that participated in the clinical trials, aren't they still being looked at?
Yes.
And in 3 years from now, TWiV will have another show on what happened to the COVID.
We're going to have a show next week.
You will know.
Listen,
I know that. But
Alan,
the answer that you're looking for is 3 years down the road.
Yes.
Alan, we don't know what the time scale is. Alan, don't use endogenization because that's meant for retroviruses.
I'm sorry.
Going into the germline.
As soon as the word came out of my mouth, I thought, oh God, I'm going to get, please don't hear that.
Letters.
So let me get
Alan just likes letters.
Let's get a little more granular. So they say the biggest question is, what's the implication of this for SARS-CoV-2? And they say, we don't know. But it's ominous that mutations that affect neutralization by antibodies are already present at low frequencies in all this, in the circulating COVID-2. And now they say, however, there hasn't been a lot of immune pressure yet because most people are not infected. And so that could be bad because as soon as there is immune pressure by infection or vaccination, it could change that, right? But they say, look, they're still, even though that's bad, it's ominous that these variants are circulating. The level of immunity needed to prevent severe COVID may be quite low, as Alan has implied. Once you get a certain amount of immunity, maybe you just get mild infections like the common cold coronas. So even if there is antigenic drift, it may not matter because it'd be mild disease in an immune population. So they
Vincent,
say, wait a minute, Dixon,
like
I'm not done. Hang on.
I'm sorry. One more thing. You were pausing.
Yeah, but you got to give me some pause. It's for,
He's got to breathe.
Margaret Thatcher used to pause and she would get interrupted all the time.
I'm watching this Crown. You're absolutely right.
Secondly, they say it's easy to update the vaccine, especially the mRNA vaccines. In fact, Moderna and Pfizer have said they could do it in 6 weeks. So and the point is we have to monitor and look at what's circulating, check them by neutralization assays against sera, and then decide, do we need to change the vaccine? So I think it's not all that bad. And as long as we're aware that we should be looking, and what we're going to have to do to look, we're going to have to sequence. Hey,
it's back to Emily.
Yes. Well, and there's already a well-established business and logistics model for updating a vaccine every year.
Yeah.
Right. We do this with flu vaccines.
Yeah, exactly.
And of course, Pfizer and Moderna can do that with their mRNA vaccines. Sanofi can do it with theirs. J&J can do it with it. Everybody who's working on one of these vaccines. Once you get through the initial clinical trials, and that was the big barrier that took a year to get, surprisingly, only a year to get past. Now you've got a fully tested vaccine platform, and you could say, okay, we're going to change 10 bases of this mRNA. It's still got the same nanoparticle around it. It's still got the same delivery mechanism, and the FDA will say, okay, demonstrate equivalence and send it out.
Yep.
And it's a much, much faster process.
So Dixon, now what were you going to say? I'm sorry. Go ahead.
No, don't be sorry. I interrupted you. So what I was going to say was what we don't have is a absolute count of the number of people that were infected with these other coronaviruses. We have an absolute count almost of the number of people that were infected with SARS-CoV-2 because it's such an important virus. How many of the other non-important nasal pharyngeal viruses of the same family did people catch before these events occurred?
Oh, how many people had to be infected?
How many total infections a year from each of these
right now, Dixon,
cold viruses
for some of these common cold coronas, over 90% of the world's population is seropositive.
90%.
90, over 90. Not all of them. There's one that is less prevalent, but some of them
That's
over 90%.
7.8 billion people.
So these infect lots of people, even you. You probably get infected every 5 years, Dixon. But most people, if you're immune competent, you don't get terribly sick. You get a common cold, and so that's why we're never going to have vaccine against 229E because Don't need
It's
it.
not an issue.
Not an
So
issue.
our hope is that SARS-CoV-2 becomes a common
It
cold.
might, and we might
Yes.
not need the vaccines anymore. Absolutely. But that may be 50
Exactly.
years from now, and I'll be gone.
Or if we do need the vaccines, then we've got these updatable platforms that have been developed
Right.
for the pandemic.
That's right.
Brienne, in 50 years, if this happens, come to my grave and talk to me, okay?
I'm not going
I
to
will.
interrupt you. I can promise you that.
I will let everybody know if I am around in 50 years. I'll give
you
you all the
please?
updates.
Brienne, who will be hosting TWiV.
That's right.
I will be a rather elderly host at TWiV at that moment, but
Or I would take that.
Dixon, you want to live to 100?
If I can live healthily to 100, yes. If I'm bedridden and don't know who I am, the answer is no.
I think that was a terrific paper. Terrific
That was
paper.
great. Yeah.
Yeah, I
Very,
agree.
very of great import, I would say. So,
Yeah, congratulations to those guys.
beautiful
Yeah.
job.
Those people.
very cool.
Actually, they have yet another very interesting preprint, which maybe we'll hit in the future. Tremendous productivity and interesting stuff coming out of that lab. I would like to go to picks now because Dixon and I have a TWIP at 4. So, Dixon, what do you have for us? Now, you're muted.
You're muted.
I know. No. I have a blatantly, no, I won't use that word. I'm plugging my cousin's son, my nephew. His name is Randy Despommier, and he's a musician. And he's about to release his first jazz album, and it's called Dioche, which will be released next month. And I've included a sample of his music, and he's got a YouTube explaining what this title means and how excited he is about this. So this has got nothing to do with virology. This has got something to do with just enjoying some cool music. And he's an amazing alto sax player, and I think he'll enjoy listening. So I just put it in there.
Cool.
Very
All right.
cool.
Very cool.
Okay.
That's
You
the
know-
kind of thing we need more of right now.
Yes.
Exactly. That's what I thought.
I enjoy virology too, Dixon.
Listen, that's why I'm here.
I don't
I
enjoy
live for this.
I don't enjoy people dying, obviously, but the virology is very interesting. Brienne, what do you
No,
have
absolutely.
for us?
So I did some searching and did not find pics of similar things in the archive, though it turns out that the word giant is used frequently enough that you get some interesting things if you start looking at TWiV and giant microbes. But I picked the SARS-CoV-2 that is made by the Giant Microbes group. If you go to their website, giantmicrobes.com, you can actually get sort of stuffed versions of many microbes and other immune cells and things like that. And I personally find them incredibly useful for teaching. And the thing that made me think about this SARS-CoV-2 giant microbe that I had is that I think that within the past 2 or 3 days, I have been sitting here on Zoom talking about spike and showing people the spike and kind of using this as a model a really surprising number of times. And so if people wanted to look at it, it can be really useful. I know that there's an antibody with detachable antigen that I use in classes all the time. I actually use a lot of the products from this site in my classes, and the students get a big kick out of it. So if people need a model to reference that's also, not quite so scary looking, these are the
This
models
is
I
cool.
tend
And
to
they do a
use.
whole bunch of medical model type stuff. I mean, you can get an intestine, you can get very cool.
They do. So I don't know if you noticed the one that my students particularly enjoy, and I know this is probably, sad to say, is the cancer cell, which the cancer cell is a nice, happy looking cell. And it unzips and turns inside out to transform to turn into a sad looking cell that has a lot of problems. And the students particularly enjoy the cancer cell.
That's good.
To understand one cell transforms into something
Transforms into another cell.
Yes.
It turns
I have,
inside out and it's a different thing.
I have many of these. People have given them to me over the years.
Didn't we throw these into the audience at a TWiV event? At some
Yeah,
point?
at the ASM. This is a rhinovirus. So, Brian, I'm interested as to what you tell them. Which protein is making up these eyes here in the SARS-CoV-2?
That is a really great question. I don't have the rhinovirus, so I don't tell them anything about that. But
yours has got eyes too.
Mine does have eyes. I don't really talk about the eyes. I have one that is supposed to be EBV. Mm-hmm. And the EBV also has eyelashes. I think it's supposed to be looking alluring, so you want to
kiss it.
It's
kiss it.
It's the kissing disease.
Yeah,
exactly.
Right. Yeah, they get a little playful there.
So, Brianne, am I correct in assuming that in the antibody antigen guide that it's a Velcro?
It's not a Velcro, but it's a little plastic tab, so you can attach and unattach. But the different parts of the antibody that I talk about in class are actually colored differently. So it's pretty easy to talk about them with the students and show the different antibody parts. I use that one all the time.
I also notice here that they have a 16-inch version of this.
They do.
They got a giant one.
Go for it, Rich.
Bonus points for giving these to your toddler.
I may have given them as presents to friends' children.
Happy baby shower. Yes.
Yeah, we did we threw a bunch out at ASM.
Yeah, a bunch of people caught viruses from us today.
Yes, so maybe the people who've been listening for a while may be aware of this, but perhaps the new listeners are less.
That's great.
So.
It's a great site.
Alan, what do you have for us?
I have, I'm going to follow along with something. Actually, there's kind of a theme going here where we just got something warm and cuddly from Brianne that you can, just curl up with. And Dixon gave us something we can listen to. This is a game. And if you don't think of yourself as a video gamer, this is still a game you might like. It is a comedy role-playing game with a Western theme called West of Loathing. It actually came out a few years ago. It'll run on pretty much anything that'll run a word processor practically. It's available on Windows, Mac, Linux. You can buy it in the Steam store. You can get it on the Nintendo Switch or the PlayStation or the Xbox or and it's an adventure game. You play as a little stick figure person who's setting out to, as a young person, to make your fortune in the West. And it is a brilliant spoof, an absolutely hilarious spoof of both Westerns and video games and role-playing games. Whether you've played role-playing RPGs in video games or not, you'll enjoy it, because if you have, you'll get the jokes that they're making about them. And if you haven't, it's a great introduction to the genre. So this is just something to curl up with and spend a few dozen hours playing along if you would rather not be watching, say, the news, which I recommend not watching these days a whole lot.
I know what I'm doing this weekend now.
There you go, Brianne.
Rich, what do you have for us?
Very quickly, this is a repick. I mentioned it a couple of weeks ago. This is the draft landscape of COVID-19 vaccine candidates from the World Health Organization. And I'm repicking it for two reasons. One, we have a number of new listeners who might be interested in this and missed the first pick. And second, they've significantly updated this so that it comes out as a spreadsheet and it can be sorted, and it's just significantly enhanced to the point where I think this may be one of the better, if not the best, resources for understanding what's going on with the vaccines.
Great.
Good. It's always good to have an update.
And it's WHO, so it's a global perspective.
Yes.
All right.
Awesome. Thanks.
Excellent.
My pick is kind of an insight article in Science by Peng Zhao and Zhongli Shi, whose name you should recognize by now from the Wuhan Virology group. And this is called SARS-CoV-2 Spillover Events. It's actually meant to be a commentary on an article in the same issue about the Netherlands mink farm outbreak. But they have a lot of interesting commentary. And, they say, well, we really need to, we can't cull every other animal that gets infectious, right? That gets infected. Like the mink they could cull, but we can't do others. So what are we going to do? Well, we have to have quarantine measures in all domestic farms and high-density animal populations, and we have to have immunization of the animals and people. And, of course, it's exactly what we said a month ago or 2 months ago on TWiV, which I like, that the TWiV recommendation is echoing around the world. Then she talks about these spillovers and back spillovers, and she talks about the possible origin of SARS-CoV-2, and she says it's very interesting. The virus, whatever it originally came from, it had to further adapt either in animals or humans before it caused the pandemic. And she says, therefore, an animal species that has high population density to allow natural selection and a competent ACE2 protein for SARS-CoV-2, mink, for example, would be a possible host of the direct progenitor of SARS-CoV-2. And she says that we need to do more sampling to sort this out. But we have to do that. And then finally, she says recently antibodies to SARS-CoV-2 have been found outside of China before the outbreak was detected. So these are retrospective analyses of sera. And they showed that potentially the virus was infecting people in other countries, not China, before we learned about it in China, which is quite interesting. And she says we should really follow that up, and we need to look in animals as well. So it's a nice little short review article. And it should be open access, so you could check it out. And then we have a listener pick from Daniel. It's an NPR article. His vaccine story inspired his father to write a Disney classic. When the classic, of course, is A Spoonful of Sugar, and it was written by the late Robert Sherman, who wrote it with his brother Richard. And the story is told by his son, who said, my dad was in a funk, and he was, I came home from school, and he said, how did the immunization go? And the son said, there was no shot. They just put some sugar in it, and we drank it. And of course, that's the polio. And from that, he got the idea, a spoonful of sugar helps the medicine go down.
Mm-hmm. Cool. I like that.
Awesome.
That's just great story. Anyway, read it. It's really good.
That's great.
All right. That's TWiV 705. Microbe.tv/TWiV for the show notes. You can send your questions and comments to twiv@microbe.tv. And if you like what we do, consider supporting us. Microbe.tv/contribute. Dixon Despommier is at Racaniello.org, thelivingriver.com. Thank you, Dixon.
Thank you, Vincent. Wonderful guest. Wonderful time.
Brianne Barker is at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove's at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thanks. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.